Hello, everyone, and welcome to the Mastermind Masterclass. My name is Candace Chapman, VP of Marketing here at Genomenon. Thank you for joining us for today's live session. Wherever you are, we hope you are safe and healthy and have showered at least once since March. This masterclass will focus on one of the latest features added to the Mastermind Genomic Search Engine, which is the ability to search by therapy. Searching by therapy is a professional feature, so if you currently have the basic edition, some of this may not look familiar to you. During the demonstration, you'll see many other features that you may not be aware are available in Mastermind, and this is a great time to get reacquainted with the full functionality of Mastermind and see if you and your organization can benefit from Mastermind Professional Edition. It's our goal to make Mastermind the first resource you use when interpreting variants, and I hope that this deep dive masterclass will help you get the most out of it. Let me cover some housekeeping items before we get started. I wonder if you're hearing the thunder that's outside. You can submit questions in the question box at any time during our time together, and we'll have a Q&A session with the founders after the demo. Today's masterclass is being recorded, and a link to the presentation and the Q&A transcript will be emailed to you Friday or Monday. All right. Let's see. Oops. Once again, we have gathered our two amazing founders to discuss Mastermind in depth. And we also have an MVP from the data science team to walk you through the demo. Introductions are usually pretty boring, so I thought we'd change it up a bit. And I asked our presenters to share something interesting that they've done since the pandemic. Our first presenter is Dr. Mark Kiel, co-founder and chief science officer. Hey, Mark. Mark has started baking and making with his daughter. Last night's specialty was homemade ice cream, which sounds really good, and I'm jealous that you didn't share. Next up is Steve Schwartz, co-founder and chief technology officer. Hey, Steve. Steve and his friend Josh built a website to help Michigan restaurants during the lockdown called saveMIFaves.org. It's pretty awesome. You should check it out, and I hope that other states are doing the same. Then we have Lauren Chun, our manager of data science and bioinformatics. Hey, Lauren. Lauren successfully moved to a new apartment and took up painting again all during this time. So enough with the introductions. Let's welcome Mark Kiel for the scientific update. Hey, Mark.
Hey, Candace, thanks. And thank you to everybody in attendance for joining us. I'm going to leave a lot of the demo to Lauren, who, as Candace mentioned, is a star employee. She's been with us now for 3 years, and she works on both quality initiatives on the software side in the clinic, as well as data project work that we do in data licensing on the pharma side. She's going to cover the demo. I'm going to return again to field any questions that you have during the course of the webinar that, as Candace said during the housekeeping, you can submit into the chat. And I'll touch on how what Lauren covers with respect to therapies, I can magnify into the overall value that Genomenon is providing for clinicians and variant scientists doing genetic testing, as well as on the pharma side for drug discovery and clinical trial design. But before I do that, let me turn it over to our CTO and my colleague and buddy for the past 6 years, while Genomenon has been accelerating its reach into the clinical market and now making great strides in pharma. Steve will describe what I mean by genomic associations and how the therapy and the therapeutic indexing that we're doing on our database is really accelerating our vision of organizing the world's genomic evidence to make these discoveries and diagnoses in pharma and in the clinic. So Steve, take it away, and I'll see everybody on the other side here.
All right, thanks, Mark. Let's bring up my slides here. There we go. All right, first of all, thank you everyone for attending. I'll just give some quick background and context for how therapies fit into the offering that we have within Mastermind, which Lauren can then demonstrate for you. So I think many know us as organizing and indexing the entirety of the medical evidence, making it easily searchable through our Mastermind search engine. I think where the real value starts shining through, though, is when we take that amount of data that we've organized and we proactively identify the genomic associations that are present within that medical evidence. And so if we go to the next slide here, I'll show you what I mean by identifying those genomic associations is that, there we go, is what we're doing is we're finding each of these types of genomic entities or concepts in the medical literature. We're finding genes, variants, diseases, phenotypes, categorical keywords, and therapies. And then we're drawing the associations between any two or more of those types of entities. So for example, it can even be within the same type of entity. Like you can plug in a gene and we can show other genes that are associated with that gene. Or you can query by variant and we will show other variants associated. On the right-hand side, you can see a very straightforward association being a disease input and a therapy output. That's great, and it gives you something to start looking into, but where the real strength of this evidence starts to shine through is when you can connect the dots with more concepts within that relationship. So not just disease therapy association, but why is that disease associated with that therapy? And in this case, being able to insert a gene association between the disease and therapy helps you to start understanding the area of the genome that the disease and the therapy both affect. And then if you can further characterize the association with a variant, now you really start understanding the functional consequence of the variant and how that leads to this disease and why that therapy may be effective for that particular expression of that disease. Or, sort of where the real power comes through is being able to fully characterize the associations, where we see at the bottom here a disease gene variant therapy association characterized by phenotypes and the categorical or clinical categories and keywords that characterize those associations. And so now with that in mind, taking a look at an example of what can be done with our API, using this association information is, you might start with the basic use case of that I think a lot of people are familiar with, but where you search for a variant and we'll show you the number of articles in the literature that cite that particular variant. What gets really interesting is if you have a specific patient, for example, or a case, you might have 3,000 to 5,000 variants that you have in that case, that sequence. And maybe you have 5 to 15 phenotypes that are associated with the patient. And our API can take a look at all of those variants and phenotypes together holistically, identify articles that cite more than one variant or any combination of variants with one or more phenotypes, and we can prioritize the articles by the types of evidence that they have. For example, prioritizing articles at the top that have the most variants and phenotypes associated with each other, or prioritizing the articles that provide the best combination of variants and phenotypes that satisfy most of what you're seeing in that case. And then once you have those papers identified, you can start to pull out other association information. For example, this graphic shows us taking those articles and then pulling out what diseases are those articles talking about. Similarly, you can also pull out what therapies are the articles talking about as well, which is something I'll get to a little later. In the meantime, I'd like to turn it over to Lauren.
Hi, everyone. I'll be doing a demonstration of the Mastermind interface and our new ability to search for therapies within it. I share my screen here in just a second. So we have this new capability to search for therapies in the Mastermind interface, and how we use that capability may be dependent on what our particular use case is. One of the main use cases could be that we know of a therapy and we're looking for associations with a gene or disease. And the other main use case is that we have a gene or disease and we're looking for associations with unknown therapies. So for the first use case, I'll be looking at an example of the drug Luxturna. Its full name is Beretagene Naparvovec, which you can see which we match Luxturna as a synonym of. When we search for that, we'll be brought to this main page here, which shows us the associations of Luxturna with any genes. And the first gene on this list is RPE65. So if I click on that gene, we'll be brought into the Mastermind interface. So there are 4 main panels in the Mastermind interface. We have first the variant diagram in the upper left-hand corner. This shows us the variant landscape of RPE65. So this is the variants and their positions as well as their article counts. Similarly, in the left-hand corner here, in the lower left-hand corner, we have a list of variants as well as their article counts. And then in the upper right-hand corner, we have the publication history. So this shows when the articles were published as well as a metric of their relevance by the journal that they were published within. And then in the lower right-hand corner, we have the actual articles that were matched to Luxturna. And so if we click on one of the articles, we'll be able to see the PubMed data. So that's the title, the abstract, the authors and publication date, etc. And then further, we'll be able to see the full text matches where Mastermind actually found the gene RPE65. Further than that, though, we can limit what we see in the full text matches by the therapy that we search for by selecting therapies only. So now you can see here, we see Luxturna matching as well as its full name. And then we also see this other synonym here, which is likely one of the first names that Luxturna was given in these early papers, which you can see up here as well in this publication chart. That the first paper we really have for Luxturna was in around 2008 in the New England Journal of Medicine. And then we have much more publications coming out around 2017 to 2020, including this very large paper in the Cell Journal. As we go through the rest of the papers, we can continue to see these matches to Luxturna as well as the gene that we search for. But the other use case in addition to this one is that we know what the disease or the gene is, but we're looking for therapies we don't know of yet. And for that case, I'll be searching for a gene called WAS, which is causative for a disease called Wiskott-Aldrich syndrome. So if I search for those two, I'll be brought back to the Mastermind interface as it was before. But here we're going to do some further filtering so that we can discover these unknown therapies. So first we'll go to this button here, Filter Categories. And we'll click on Clinical Significance, and we can see that we can limit our results by keywords related to diagnosis, prognosis, therapy, or clinical trials. So for this use case, we'll be looking at the therapeutic keywords. So you can see all of the keywords we have here, things like antagonist, therapy, or inhibitor. That's going to show us all of the therapeutic information related to that disease and gene that I entered earlier. So we're going to enable all of these keywords so that our results will be limited by all of those, and we're going to submit with chosen filters. And essentially what this does is it filters out the irrelevant information, aka non-therapeutic information. It prioritizes the info that is related to therapy based on all of these keywords I've selected, and then displays that information to show us exactly why that information was prioritized. So we can see here that the first article we matched here is in the journal Blood, showing the effects of Altrombobag. So that's the first therapy that we're noticing here. And in the full text matches, we'll see that there's an empty box. And because we're showing therapies only, which is just the therapies that we've searched for, like we did with Luxturna. So what we need to do is just change that to keywords only. And then we'll start to see all of those keywords that we selected earlier, including things like agonist or treatment. And then you'll see that Altrombopag occurs next to treatment, so it's an easy way to discover therapies that we didn't know about before. The next article shows even a gene therapy, where we matched, for example, hematopoietic stem cell gene therapy in the text. And it goes even down to showing articles like clinical trials, where we match things like dose escalation, as well as more general things like interleukin-2 therapy. With this particular use case, though, when we're looking for unknown therapies, we may want an actual list of the therapies rather than just this list of articles and the full text matches, in which case we can achieve that through the Mastermind API. So we'll go to the API demo page here, where we can try out the Mastermind API. So since we're looking for unknown therapies, we're going to start with the therapies endpoint. The first thing that we need to do is retrieve the API token, which you'll need a Mastermind account to do. So you fetch the API token, and then you're able to enter in a gene. In this case, I'll be demonstrating the gene BRAF. Once you submit the query, you'll be able to see all of the therapies that have matched in association with BRAF, prioritized by the number of articles that they matched within. So you can see things like vemurafenib, cetuximab, or cisplatin, and the number of articles in which they matched associated with the gene BRAF. These API endpoints, however, don't exist in isolation, so you can use them in combination. So I can also paginate through these results to get a complete list before I do this. See other therapies like erlotinib and trametinib. So if I take the ID for erlotinib, for example, and I want to know what articles erlotinib and BRAF were found within, I can take that ID and go over to the articles endpoint. Once I enter in the uni ID there, as well as the gene BRAF that I had before, I can submit that query and be able to retrieve all of the articles here that match to BRAF, as well as that therapy erlotinib. So all of these API endpoints can be used in combination depending on your particular use case, and that's described even further within the full API documentation. And next, our CTO, Steve Schwartz, who you heard from earlier, will be describing how to combine multiple endpoints and organize that data in a custom format. So I'll pass it on to Steve.
Awesome. Thank you very much, Lauren. Let me show my screen here. All right. So one of the endpoints in the advanced API that really helps do this kind of analysis that I'm about to show is the article endpoint. So if I show the same, if I take one of those PMIDs, for example, where Lauren ended with showing the list of PMIDs that were associated with that disease and that gene, that therapy in that gene, then you can plug it into the article info endpoint, any PMID here, and we show you everything that we found in that given article, meaning every disease that the article talked about, every phenotype, every therapy, every gene, and every variant. And so taking that information and plugging it into our own Python script that I've written that I can demonstrate here, we can pull out a lot more information and do a lot more sophisticated analysis with that using the API. So I have an example script here that finds newly matched evidence for one or more genes, and then additionally looks through the information that the advanced API returns for those newly matched articles for that for the list of genes that you have, and it pulls out all of the therapies, and it also will score the articles in a prioritized order of what I like to call interestingness, or which articles are probably the most valuable to look at from a therapy focused context. So in this case, I am going to show my test.txt file that I'm passing into my script here is a text file that has a list of genes that I'm interested in. In this case, my test file just has one gene, but you run it and it just simply starts querying the API and aggregating the data so that it can do analysis for that. Rather than make you sit through this, I think this script takes maybe about 60 to 80 seconds to run. So rather than sit through that, I'll go ahead and show you the results. So it outputs a CSV file that is a list of the newly matched articles for that gene. I use the gene ATM in this case. And so this is a list of articles newly matched for ATM with a focus on therapies. And so you can see here, I've got deep links into Mastermind so that you can click any one of those links and see the full text sentence fragments for what we matched in relation to the ATM gene. But I also just directly output the journal, the title, and the publication date here. If you scroll over, you can see the other genes that were talked about in the article, the gene variants from my gene ATM here, as well as variants from other genes in the article. And then if I scroll all the way to the right here, I have a list of therapies that were matched in each article. And then I have this weighted score that I've developed that takes into account things like how many of my genes were matched in the article versus other genes that weren't in my input list. So this score kind of gives me an idea of how much the article is about my gene versus just mentioning my gene among many. And then I do the same thing for variants in the article from my gene versus other variants from other genes. And then I give additional points if they mention my gene or variants from my gene in the title itself. I also give points for if there are therapies mentioned in the title, and many other parameters that I've programmed into the script here. And so what's really interesting here is if I take my aggregate score and I sort in descending order, I should get more interesting articles at the top that make it easier for me to go through and investigate each article in turn. So you can see here, based on my my quick therapy focus scoring here, that I get articles at the top that talk about the genetic susceptibility to melanoma in gene panel testing, or the role of genetic variations in hepatic handling of drugs. So you can see I'm getting articles talking much more about functional mechanisms and therapies at the top here. But of course, this score can be can do anything you want. This is what I've built in the Python script. This is just sort of the quick scoring system that I came up with. We do make these scripts available as open source in our Mastermind API cookbook repository on GitHub. And so there is a lot more examples in that repository, and you can sort of use it as a jumping off point to really start to explore the power in the advanced API. So I think I will turn it back over to Candace now.
Thanks, Steve. Appreciate it. Thank you, Mark and Lauren. Lots of great stuff. I want to remind you, you can put questions in the question box there. We have a few coming in, so let's get to some questions. Here we go. My mouse has not been acting properly. All right. How can you prioritize references if you aren't yet sure what therapy you are looking for? Who would like to answer that?
I'll take the UI, Steve, and you can clean up the answer with the API, which I think offers a lot more flexibility. And I like the coining of the phrase interestingness. I think you should trademark that. And I'll emphasize that what Steve described in his discussion of the script he made is extremely flexible and modularizable. And so Steve settled on some parameters for his interestingness score that satisfied the question that he was seeking to answer, but that can be permuted in any way that you please. So on the UI side, what Lauren showcased in the article list is by default organized by relevance. And if you remember the icons in the publication plot in the upper right, the size of the icon, what Lauren referred to as a big article, meaning it has the most relevant and interesting content, and that's reflected in the size of the icon. That relevance takes into account how much of the content focuses on the therapies, and particularly how much of that content focuses on the therapies with respect to treating that genetic causation. And so relevance is a parameter that's built into the user interface and can be set as, it's set as the default, but it can be set to showcase the most relevant articles to your search up front. An additional way that you can prioritize the articles would be by publication date. So depending on your concern, you may be aware of therapies that are useful for treating genetic disease that's been known for 30 years, but you're interested in the latest, either a review article or the latest things like gene therapies that Lauren highlighted in her examples. That can be brought to bear in the prioritized article list by modifying the sort parameter from relevance to publication date. And then the last thing I'll say is forward-looking to a user interface feature that we're building out now in professional, is to showcase some of those association data that Steve alluded to in his diagram and that Lauren highlighted in her demo of the API. Some of those associations will be put to a page in the user interface for professional, so that you'll be able to see some of that itemized content for specific therapies that are recurrently associated with different genes and diseases. So, Steve, I'm sure that there's more territory that you can cover there with respect to the API, but that covers a broad swath of the UI capability.
Yeah, absolutely. So from a high level, if you think of any given investigation or any given search, the way that the data is presented that we have, it's basically a web of interconnected associations between the 6 different concepts that I had described earlier: the gene, variant, disease, category, keywords, phenotypes, and therapies. And so I think each search, it's really about figuring out what your constraints or your input values are, and then deciding what you're looking for as the output values. The user interface currently really helps you get to the articles as the output values, so that you can then look at each article and decide what you're trying to get from it. And we try to pull that information through in the interface to make it easy to get to. What's really interesting with the API is we have different endpoints for each different set of outputs. So we have an endpoint where whatever your input values are, we will list associated genes as the genes endpoint. Or for those same sets of inputs, give me a list of variants from the variants endpoint. Or again, from the same set of inputs, give me a list of therapies or phenotypes or articles. And so that's one of the ways that the API makes it much easier to constrain your initial search however you can, even if it's only with one input parameter like a gene. You can use that gene to list out the associated therapies. And then what you can do is you can loop through each therapy and use that each therapy as an additional endpoint. So for example, gene and therapy 1. And then as your output, maybe hit the articles endpoint, or perhaps hit the variants endpoint to see, if there's a specific region of the gene or types of variants that you're interested in effective therapies for. And so it's really about figuring out how you can constrain your input in order to get a list of a list of options in your output, and then you can use each of those options as an additional input to further constrain your search. And so I think the first part of it is figuring out how you can sort of pivot your query to filter and refine. And then once you've gotten the refinement that you were after with the set of filters that gets you to the information that you're looking for, only then does it become a question of how do I now prioritize the information that I'm looking at. And so that's one of the ways that both the user interface and the API prioritize, we do a few things on the prioritization that are irrespective of your search. Things like looking at the recency of an article, or the impact factor, or how much genetic content is in the article. And then there's a second dynamic piece to the prioritization that we do on the fly based on your particular query, where we try to weight articles that talk more about the specific things in your query to the top, and how those things are related to each other. And so that's where, when Lauren showed in her demo, using the categorical keywords to filter for therapies, it's not only filtering, it's also reprioritizing based on articles with more therapeutic content. And so I think those two things together help to sort of first refine and then prioritize the results, to help you find what you're looking for, even when you don't necessarily know what it is yet.
Okay, great. Another question, are nutritional therapeutics included in the drug ontology?
Steve, I'll let you take it, because you're better aware of the content of data that we're using.
Yes, they should be. We use the FDA Unique the UNII, the Unique Ingredient Identifier database from the FDA. It includes both pharmaceutical therapies and nutritional therapies, I believe. I know that we've searched for a few of those and found all the ones that we searched for. So I think it would be a matter of trying it and see if it works. If you do find something that's not in there, we do have the ability to modify and add to it. So let us know if you find anything that, if you type it into Mastermind and it doesn't come up as a therapy option, please reach out to us through the contact form and let us know.
Agreed.
I know there's over 180,000 therapies in there, so lots of wonderful stuff to play with. Let's see, just got another question here. Is it possible and much easier to search for newly matched articles which show functional studies associated to a particular variant?
It is easy to, I don't know if what we're talking about easier than, it is easy through the API.
Therapies.
What's that?
That's what I'm assuming. I'm assuming she means now with therapies, is it easier?
Oh, right. That makes sense. Yeah.
Reiterating what Steve said, but emphasizing that searching in combination for the given keyword or ontologic term in our therapies index, in addition to, in combination, adding any of the functional categorical key terms. So Steve highlighted the ability to perform these searches in a multi-parameter search or Boolean search. That capability is possible also in the UI. So I think the question when she says is easier, now that we have therapies, you are definitely able to combine a therapeutic term from the uni ontology with a functional category, and then have all of those functional, empirical, in vivo, in vitro studies that address the efficacy of that therapy in the context of those functional studies. So I'd encourage you to combine the new therapeutic ontologic capability with some of the pre-existing categorical search capability that we have. And those references, if you stick with the relevance sort order, should rise up to the top, and you should see in the top 10 articles the most relevant content to that functional empirical evidence around a specific therapeutic.
Cool. Can you filter by biological pathway?
Steve, you want to take that? Because I just ate up that answer.
Sure. Not directly, not yet. One thing that you can do, one of the ways that I've done that in a couple of the scripts that I've written, is by starting with either a set of genes that I already know are in the pathway and passing those in as a group of genes into the API. Or if I don't know exactly which genes I want first, what I'll sometimes do is you can either start with one known gene or even start with a disease, for example, and query the genes endpoint of the API. So for example, if I query the genes endpoint, it's going to give me a list of genes associated with whatever my input is. And so my input could be a disease or it could be a known gene. And very often the top genes that come back in those associations are the other genes in the pathway, because those are the ones that tend to be most highly associated in the medical evidence, in the evidence of the medical literature. And so I will usually would put that as sort of a pre-function in my scripts to, start with a disease or start with a gene. The first thing I'll do is hit the genes endpoint, get the associated genes, and then I can do the rest of my analysis with the set of genes in that pathway. That way is one way to do it.
One thing to add on to that Steve showcased with respect to the interestingness parameter that he devised, which is still in that script, can be repurposed or modified or used as is the idea that not only do we find these associations, but we afford the ability to quantitate the strength of those associations based on that evidence. So it can be as simple as enumerating the number of papers citing that association, or you could go into much more specific depth by qualifying the nature of that evidence using the article's info endpoint. It's really hard to overstate how flexible the API allows you to answer specific questions and then do so iteratively. So answering a question like, is it possible to search on pathways, just as Steve described, and in any other permutation that you'd like to address. Steve talked about knowing the pathway a priori, but also discovering the pathway, whether it's direct and canonical to a pathway of interest or indirect. Mastermind affords the ability to recover that evidence as well as discover new evidence, particularly using the API as it allows you to do things in very high throughput.
Okay. So because it's just us, Mastermind community, I'm going to throw in a bonus question of my own, Mark. We've been talking about some exciting stuff around what Mastermind could possibly contribute to our current situation with COVID-19. There's going to be some stuff coming out about it, and we're really excited, but you want to just give us a little taste of what Mastermind can contribute?
Yeah, sure. Our focus is not directly on infectious disease. So when people think of viral genetics or bacteriology and parasitology as it has bearing on human disease, it's facile to think, well, let's look at the genetics of those different organisms. So we don't cover that content. But Mastermind does play a role in helping understand the host response to in this case, a viral infection. So one of the most obvious things is the viral receptors on human cells, which are now widely known to be ACE2 and TMPRSS2, widely expressed throughout the body, which is probably an explanation for the not just respiratory manifestations of COVID infection. One thing that piqued my interest as soon as information was coming out about COVID, as I'm sure it did any geneticist or physician for that matter, was the idea that there are some patients who are asymptomatic, and not properly patients, but rather carriers. There's clearly risk factors associated with morbidity and mortality associated with infection, which isn't surprising given compromises to underlying health. But the thing that was most intriguing was the idea that there can be asymptomatic carriers. And my first thought was, well, what genetically sets those carriers apart? And based on what I described as the host viral receptors, ACE2 and TMPRSS2, it's logical to think that there may be polymorphisms in those two human genes, proteins, viral receptors, that confer a resistance to infection or a diminishment of the consequences of that infection, or otherwise outside of the viral host direct interaction, any aspects of human immune biology is susceptible to being highly polymorphic across different individuals. And in the case of COVID infection, it seems to be the case that the exuberant immune response is actually what leads to patient demise. And it's quite reasonable to presume that there's some genetic risk or polymorphic differential response in association with COVID infection. And as Steve and Lauren highlighted, and I touched on in some of my answers here, Mastermind has a superabundance of this association data, not just for ACE2 and TMPRSS2, but for any other genes associated with the host viral response. And so things are preliminary at this point, but we're working in this vein with a number of the sequencing efforts that are involved in trying to divine what may be responsible for this differential response. So very long-winded answer. Yeah, we've got a lot of stuff we're doing.
And I would add, additionally, especially with the latest information on therapies that were and therapeutic associations that we're pulling from the literature, it's now much easier to stay up to date with the absolute latest evidence for any given therapy. And so that's, I think, another thing that will sort of come to the top in addition to looking at the genomic predispositions of patients or carriers and how they react to the virus, but starting to look at therapies that could be effective as well.
Are known or not yet known, as Steve highlighted with his script, right? Pulling out the stuff that's known and asking questions about those things, but also discovering things that may not be on the radar of pharma researchers.
Yep.
Awesome. Oh, well, thank you guys so much. I'm looking at your pictures here, but I know the camera's here. Thank you so much for joining us today. I hope you learned things that you can use in your daily work. And our next Mastermind Masterclass is on the essential ACMG AMP criteria filters. So I hope that you're excited about that. Follow us on Twitter at Genomenon and look for an email with details coming soon about that. We would love for you to share your feedback with us, and you can do that by going to genomenon.com/feedback. And if you'd like to talk to a member of our team about upgrading to the professional edition or have questions about purchasing multiple seats for your lab, email us at hello@genomenon.com. And thank you again for joining us. We hope you join us for our next Mastermind Masterclass. Enjoy the rest of your day.